Hi, my name is Sage, and today I will be presenting on Paragonimus Westermani. Here on screen, you will see a P. Westermani egg under the microscope. They look like little coffee beans, and they're one of my favorite parasites. Taxonomic classification. So they're under the kingdom Animalia, and their phylum is Platyhelminths, which basically means like flatworms. They're trematodes, and the genus is Paragonimus, and the species is Paragonimus westermani. Where are they found? They're found mostly in the fresh waters of Asia and Africa, and they can also be found in the Americas. There's actually not a lot of cases in the Americas. A lot of people from the Americas can get sick, but that's usually because they travel over to Asia or Africa where they do eat raw freshwater crustaceans as a form of delicacy. And so here you can see on the map where they're most prevalent. Nutritional requirements for these parasitic worms, they mainly leech off of the host's bloodstream. They break down red blood cells. They target the protein hemoglobin. And what they do is they'll use aminopeptidase to break down the hemoglobin into peptides and free amino acids, which they then take to power their own protein synthesis mechanisms. What do they look like under the microscope? So over here, the pink blob is the adult under the microscope. If you can like see right here and right here, those are like the main reproductive organs, and then there's like straight down is the uh excretory. It's like the digestive tract, and these are like the eggs, and yeah, they have a lot of space. Like most of their body is used for used to house their reproductive organs. These worms are actually hermaphroditic, which means they hold both male and female sex organs. They can reproduce sexually or asexually, depending on whether they're diploid or triploids, which I think is pretty cool. And then over here, this little circle thing is the metacarcae. Yeah, it's the infective stage of the worm. This is like the larvae form, but it's in this like protective little shell so that it can withstand the human digestive system, especially the stomach acids. So what happens is you'll eat like raw freshwater crustaceans, so usually crabs that and then that has these little infective larvae in them. Your body will break down the crabs, and they won't break down these little like larvae, and those will pass through to the small intestine. And in the small intestine, they will excised from their little shell, and then they can start like digging into your cellular walls. So their life cycle, like I said before, you'll find the eggs in the water. The eggs hatch in the water, and then they go and infect like a freshwater snail, and from there they turn from sporocytes to retiae to cacariae, and then the snail gets ingested by the crustacean, and then the crustacean get ingested by the humans, and the humans, the little metacarcae like enters the small intestine. It breaks open, starts eating the small intestine wall, and then it travels all the way to the lungs. And for the humans, you can find it in the phlegm or in the feces. So the first intermediate host is freshwater snails. The second intermediate host is freshwater crustaceans. So think like crab, crawfish, shrimp, I guess, if you could find shrimp in freshwater. And the final host is human. We have paratenic hosts like wild boar, minks, anything that would eat freshwater crustaceans. So, because there is such a large array of parasitic hosts, it is hard to get rid of this parasite. Yeah. Pathogenesis. So, the metacarcariae, however the heck it's pronounced, exist in the small intestine. So, it breaks open out of this little shell. It migrates through the abdominal cavity towards towards the chest cavity. Basically, it like travels through the diaphragm to get to the lungs, and then it becomes adults in the lungs. The adults incise themselves in the lungs, and they like form granules, something like that. Like when you look at a lung X-ray or something, or CT scan, you can see just like you could see them very clearly because it would be like dark, or would it be white? Either way, it will stand out. The adults gain nutrients by degrading the host globulin. They reproduce in the lungs, and the eggs are either passed in the phlegm, or the phlegm is like swallowed, and the eggs are passed in the fecal matter. Virulence factor. One of the ways that invades the immune system is by using a protein called paramycin, which is a structural protein that controls muscle contraction. But once like the worm enters, it becomes the paramycin has a secondary function where it becomes immunogenic, and it basically causes a host immune response. So it can regulate the host immune response by inhibiting the classical pathways of the C1 function. And to do this, it does by binding to the collagen of the C1q, which acts as an inhibitor. Scientists also think that there are FC1 receptors on the surface that binds to human immunoglobulin, immunoglobulin 1 and immunoglobulin 4, which activates the host to like protect the worms from being, I guess, attacked by the immune system. And by doing so, it induces allergenic reactions within the host, and it's associated with the Th2 immune response. During the Th2 immune response, you will find heightened levels of immunoglobulin 1 and immunoglobulin 4. Mechanisms of infection. So one of the main mechanisms is host tissue degradation. The worms do this by using a cysteine protease, which is a proteolytic enzyme. Basically, it breaks down like the host's cellular matrix and stuff. The main ones are 27 and 28 kDa. What they do is they break down like the macromolecules and the host's extracellular matrix. 28 kDa plays a vital role when the parasite's actively migrating through the host tissues, because it is the one that causes the most damage to the extracellular matrix, and also the one that degrades the fibrillar macromolecules the most. So for fibrillar macromolecules, think like collagen, fibronectin, and the laminin. Epidemiology. So who is at risk? People who eat raw or undercooked shellfish, freshwater shellfish, is at risk. Circumstances, basically, if you're traveling and you go to like a local, well, if you are traveling and you eat delicacies that consist of raw freshwater shellfish, then there's a very good chance that you might get it, especially if you're traveling in Asia or South Africa, because those are the areas that have fresh water shellfish raw as like a form of delicacy. Another way you can get it is by undercooking freshwater shellfish. So, moral of the story, make sure your shellfish is properly cooked. How to prevent it? Cook your shellfish properly. More sanitary handling of the contaminated crustaceans is also useful so that we can stop the spread of it. And not a lot is known about this. So more research and more outreach to the local communities where these delicacies are consumed would be great in stopping this parasitic infection. Diagnosis. So how do you know you have it? Well, one of the main forms of identification is by finding the eggs in your phlegm or in your poop. Serological tests can also be done, but only really for early infections or atopic infections. So infections that go elsewhere to the body that's not like in the lungs. The eggs are golden, yellowish brown. Like I said, they kind of look like coffee beans. They have a really thick shell, as you can see, and then they have a very obvious operculum, which is this little like hinge thing. It actually opens up like a hinge to let the larvae escape once it's in the water. How do you like? What are the symptoms? It's a cough, rusty slash blood tinged phlegm, pain or tightness in the chest, difficulty breathing, or recurring hematitis. For chronic infections, fever, anemia, weakness, and weight loss. One of the main telltale signs of a parasitic infection is that if you're eating a lot, but you're not gaining any weight, or if you're losing weight, but you're still eating a lot, it's usually because like the parasite leaches the nutrients. And so you need more to have the same amount to function. It can attack the central nervous system. Like I said, the parasite can go elsewhere in the body, so it's not the lungs. And when it infects like the brain or the spinal cord, it can cause headaches, seizures, and visual disturbances, along with the rest of the symptoms. How do you treat it? Praziquantel is the tried and true method. It's usually what is normally given. You will For kids and adults, it's 25 milligrams per kilogram given orally 3 times a day for 2 days. How praziquantel like kills the worms? It causes severe spasms and paralysis of the worm's muscles. It does this through the rapid influx of calcium ions through like the um, worm's membrane, and which basically causes the worm to seize up. It is ineffective against juvenile worms, though, so it only really kills the adults. Triclobendazole is another medication that works against these parasitic worms. And for this one, it's 2 doses of 10 milligrams per kilogram, given 12 hours apart in patients 6 years and up. And this kills the worms by causing a reduction in the resting membrane potential. Kind of the same thing as praziquantel. It basically causes like mass spasms and paralysis. But for triclobendazole, it also inhibits tubulin function and protein and enzyme synthesis. So the worm can't do what it needs to survive, basically. For drug susceptibility, some patients can be allergic to praziquantel. Triclobendazole does not work all of the time, and when trizobentacol doesn't work, you need to treat it with praziquantel. And if praziquantel doesn't work, then you have to treat it with triclobendazole. And if both are ineffective, what one case study did was they desensitized the patient to praziquantel, and that was how they treated their infection. For drug resistant, as of right now, there is no known drug resistance for this parasite of the 2 drugs that work. However, since there are only 2 drugs that work, it is really important that there is more research going on to try to find out more about this parasite and the disease and how it works. There has been research put forward to developing new drugs and new diagnostic techniques, which is very important because Paragonimus is easily mistaken for tuberculosis or lung cancer. And there are so many proteins and, I guess, mechanisms that drugs can attack. So there is a lot left to be studied. But yeah, That's it. They're really cool parasites.